All right. Again, apologies for the late Friday here. Last year, when we did the late lectures, the Giants were in the playoffs, and so I was like telling half the crowd what the score was in the games, trying to keep them somewhat interested. If you care, I have the scores of the baseball games today, but most people probably don't care. Milwaukee beat Arizona, if you're interested. So what we're going to do today is, oh, what happened here? Oh, no. Projector decided to go off. Should come on in a second. Give me 1 second. All right, so we're going to talk about endosymbioses mostly today, and then in the next lecture about extremophiles. I'm going to finish up, I know it was painful, but I'm going to finish up the crazy tour of eukaryotes because these lineages are worth knowing about, and we're going to talk about them a little bit. And the last, I think the one lineage I did not cover earlier was the unicont lineage. Is that true? I did not cover this one. Not that anybody remembers, but I did not cover this lineage. So this lineage includes many organisms people care about, like us. There are 6 major branches in this group. I'm not going to talk about these two here, the fungi and the animals. You're going to either really love them or be very sick of them later in the course. There is one lineage that I am going to talk about, which is the choanoflagellates. This is the sister lineage to animals. So they are more closely related to animals than animals are to fungi or than animals are to anything else. There is a lot of research in this group of organisms to try and understand animal evolution by studying, in addition to animals, the sister group to animals. And there are some really interesting properties in this group. Don't know if any of these, I don't think I have any videos in this group, but this group includes a lot of organisms that form these sort of simple colony structures. And many of the cells in these organisms resemble some of the complicated cell structures seen in a variety of animals, such as sponges, and you'll hear about these later in the course when we talk about animal evolution. But it's just important to realize that there are some lineages out there of microbial eukaryotes that are reasonably
Arrows indicate the captured food items that are about to be ingested. This choanoflagellate Salpingica species captures and swallows food. This sequence is shown at real time.
They basically all live sort of like amoebas. They have these sort of flexible cell structures. They have these things called lobe-shaped pseudopods. They feed by something called phagocytosis.
These more highly magnified sequences are shown speeded up 25 to 50 times. Arrows indicate the captured food items that are about to be ingested.
Of many eukaryotes, many eukaryotic lineages, and this ability to form sort of very complex cell structures is a hallmark of eukaryotes, probably related to component of eukaryotes that we didn't talk about before called the cytoskeleton. It's yet another one of those things that appears to have evolved in the branch leading up to the common ancestor of eukaryotes, like the nucleus, like splicing of RNA, etc. These amoebozoans form, again, these sort of blobby structures. This includes this one that I showed the video of before. They can crawl across surfaces. They can form these sort of semi-multicellular structures.
An amoeba moves across the field at 25 times real speed. This cell has eaten numerous green algae, now contained in digestive vacuoles. Movement is generated from cytoplasmic streaming by actin microfilaments.
Fruiting body structures. There are a lot of different lineages of eukaryotes, I think I mentioned this before, that are multicellular. It shows up many different times in different branches of the eukaryotic tree. Appears to have been each time a separate invention of multicellularity. these are, we microbiologists like to claim that they're the basis for things like the blob, but
this acellular slime mold,
so I want to spend a couple minutes talking
is a
about
single
the
cell
midterm,
enclosed by the cell.
And then we're going to go into endosymbiosis.
The cellular slime
so the
mold,
midterm is going
Dictyostelium
to be
discoideum, has 4 different life
questions,
stages. At this vegetative life stage, unicellular haploid myxamoebas move independently from one another, engulf food particles by endocytosis, and reproduce sexually and asexually.
And some short answer questions.
When conditions become unfavorable, myxamoebas aggregate and form pseudoplasmodium, or slug. The myxamoebas retain their cell plasma membranes. The aggregation in this sequence took place over about 2 hours. Slug formation took an additional hour. Sequences of the straggler amoebae are shown speeded up about 25 times.
So Brian Moore was originally going to give lecture 13 before the midterm, and we thought that that was going to be a little confusing to introduce a new topic and then have you tested later in that day on a previous topic. So I'm going to do that lecture and do a little bit of overview of some of the microbial stuff, as well as introduce a couple of new things in there. We've scheduled some review sessions. I still haven't had time to confirm with the registrar's office exactly when they're going to be, but I'm pretty sure that we're going to have a review Monday afternoon after the 3 o'clock class, so from 4 to 5, and then Wednesday afternoon, probably after the 3 o'clock class, and Thursday late afternoon, early evening. And I'm also going to be available in office hours on Monday, and we might try and schedule some extra office hours too. And I'm going to try and send around at least some practice questions, if not sort of a full-fledged practice midterm that will be about the length of the real midterm. I'm going to try and send that around this weekend. Are there any questions about the midterm? You should, yeah. You should bring a pencil or pen. A pen is better. Oh, I just changed the slides. So a pen is better because for the short answer and diagram questions, if you disagree with our grading of them and you want to request a regrade, you can't do that if you've answered in pencil because it could be changed. So we're only allowing sort of requests for reevaluation of things if you do the short answers in pen. But we will provide the Scantrons for everybody, so you don't have to bring a Scantron. You have to bring a picture ID in order to get the Scantron that's assigned to you. So you should go, you'll get an email saying where to go. I think for this section it might all be in here. I'm not 100% sure, but there might be another room and that'll be alphabetical. So you should go to the place where you're assigned in the email because that's where your test with your Scantron will be waiting for you. Okay, so what I want to do now is talk about one aspect of this acquiring functions from other organisms. So if you look at evolution of organisms, organisms invent novelty. That's what a lot of evolution is about. They evolve a new structure, a new biochemical pathway, a new feature. And one of the ways that microbes participate in this is very interesting in that a lot of eukaryotes have figured out that they can acquire new functions by bringing microorganisms either inside of them or into some sort of partnership and utilize the functions present in those, frequently bacteria, for themselves. So I mentioned before that there's an incredible diversity of metabolism in bacteria and archaea, not so much in eukaryotes. Eukaryotes take advantage of bacteria and archaea by engaging in these symbioses with them. And I'm going to talk about 2 critical symbioses in the evolution of eukaryotes today. And that relates to these 2 organelles, the mitochondrion and the chloroplast. So organelles are formally any membrane-bound... Hang on a second. You'd think they'd have rechargeable batteries at Davis. Or batteries that even work. Do you know how to do this? Let's just try it the other way. If this doesn't work, we might... Ah, right. Or professors that know how to put in the batteries. All right, so organelles are formally any type of membrane-bound compartment inside the cell of an organism. And you do see things that you could call organelles across all types of organisms, even in, occasionally, very rarely, in bacteria and archaea. These two organelles in particular are really interesting in eukaryotes. So eukaryotic cells have the endoplasmic reticulum, the Golgi apparatus. The nucleus is an organelle. But these two in particular, the mitochondrion and chloroplast, are very interesting because if you look at them, as people have done now for hundreds of years, they look a lot like bacteria. Or, you know, people didn't know what archaea were before, but they also look a lot like archaea. They're frequently surrounded by many membranes, even more than, say, the endoplasmic reticulum. The inside membrane in them frequently looks physically like the inside membrane on many bacteria. They morphologically, just their shapes resemble bacteria. Many of them contain DNA inside of them. They have their own genomes. Many of those are circular pieces of DNA, like we see in bacteria and archaea. And many of the internal components inside these resemble internal structures seen in bacteria and or archaea. So for example, chloroplasts have these layers of membranes where photosynthesis happens. You see those same types of things in many photosynthetic bacteria. So people have wondered for many years if mitochondria and chloroplasts are evolutionarily derived from bacteria somehow that are living inside the cells of eukaryotes. Very important related to this, in some of the early studies of chloroplasts, people found that some chloroplasts had peptidoglycan around them. Remember what peptidoglycan is? How many people think peptidoglycan is in bacterial biofilms, is A? How many people think it's a component of the archaeal cell wall? How about the bacterial cell wall? Yes. Yay. How about a pigment used in photosynthesis? All right, so peptidoglycan is one of the features that I talked about as being widespread and unique to bacteria. It's not seen in archaea, not seen in eukaryotes. This is really important for studying evolution of chloroplasts. So if we ask, you don't have to read that whole question, I can just explain it to you. If you ask, what features of chloroplasts, scratch out the mitochondria in that question, are good evidence that chloroplasts are derived from bacteria? They contain their own DNA. That's not great evidence that they're derived from bacteria because archaea and viruses and eukaryotes have their own DNA. They look like bacteria in shape and structure. That's not great evidence because, as we've told you, many archaea look like bacteria in shape and structure. The fact that some contain peptidoglycan in their cell envelope was a smoking gun, that some of them were probably derived from bacteria, because peptidoglycan was a hallmark feature only seen in bacteria. And carrying out important metabolic processes, archaea and some microbial eukaryotes could do that too. So all of these things suggested that they had been derived from other organisms. But the peptidoglycan was like the first evidence that they probably were bacterial in nature as opposed to anything else. Now, conveniently, there's another way to study the evolution of mitochondria and chloroplasts. They have their own DNA. So you can take that DNA, extract it, read the sequence of letters in that DNA, and compare it to the DNA from other organisms. And fortunately for us, many chloroplasts and mitochondria in their genomes have genes for things like ribosomal RNA. So just like Carl Woese and others built evolutionary trees of all organisms on the planet by comparing sequences of ribosomal RNA, we can now include in those same trees mitochondria along with everything else and ask, where do they sit in those evolutionary trees? By looking at their own DNA, just like we did with building the three-domain tree of life, or studying the evolution within bacteria, archaea, and eukaryotes. So inside a eukaryotic cell, there's a nucleus, and inside the nucleus is what we will call nuclear DNA. Inside the mitochondria is mitochondrial DNA. And we can study the evolution of, in essence, all the DNA inside the mitochondrial genome, but fortunately there are some of these ribosomal RNA genes and other genes that are useful for studying their evolution. So before we do that, I'm going to show you what that looks like in a minute. Even without doing that, we can create a model sort of in our heads for the evolution of mitochondria and eukaryotes. And the way we can do this is if we look across this ridiculous number of eukaryotic groups that I told you about, even if we look at the major lineages, all of them have mitochondria, except these two I mentioned before, the diplomonads and the parabasalids. Everything else has mitochondria. The simplest model here is to suggest that the common ancestor had mitochondria, and it was then lost in these lineages that don't have it. Or since they appear to be sister taxa, was lost once in the lineage leading up to the common ancestor of those two that do not have mitochondria. So this is one of the reasons I walked you through all those different groups, because these two in particular that don't have mitochondria are really important for understanding some aspects of mitochondrial evolution. Did it kick out again? Or no. But just from this pattern, the presence of mitochondria, we can use our character state reconstruction methods to hypothesize that the common ancestor probably had mitochondria. So when people have done that, they have then asked sort of more refined questions. Are mitochondria in fact derived from bacteria or not, as they physically seem like they might be? And how many times did they originate? So it is possible that the mitochondria were present in the common ancestor of everything, but maybe there were multiple distinct mitochondria then, and different lineages inherited different forms of them. Or maybe that model that I told you from looking at this tree is wrong. Maybe each of the lineages that have mitochondria separately acquired mitochondria from some type of symbiosis. So we can study that again by looking at the DNA inside the mitochondria. And if we build an evolutionary tree of that DNA, this is an example of what one of those trees looks like. I haven't shown it on here, but an outgroup to this group here are archaea and eukaryotes. Mitochondria, when you build trees of their genes, show up in the trees inside one of the bacterial branches in the tree of life. That says they are, in fact, evolutionarily derived from bacteria. They are embedded phylogenetically within the bacterial branch of the tree. In fact, they are in one lineage of one of those 6 lineages that I told you about of bacteria. So they are inside the proteobacteria, and in this one group of proteobacteria called the alpha proteobacteria. So of all the distinct lineages of bacteria, mitochondria came from one of them, the alpha proteobacterial subbranch of the proteobacteria. So if we now look at the full evolutionary tree of bacteria, which I've not really shown you before, I've shown in here like 30 different phyla of bacteria, or maybe 40. They're actually thought to be about 100 major lineages of bacteria. We only talked about the proteobacteria, somewhere down here, the chlamydia, the spirochetes, and a few of the other lineages. So we're not going to go into all the detail about all these other ones, but mitochondria came from this one lineage, the proteobacteria, and from this one branch within that lineage, the alpha proteobacteria. And if we look at this tree again, these are all, sorry, I didn't say this. These up here are all, in essence, mitochondrial genes. They all share a common ancestor with each other to the exclusion of all other bacteria. There was one event in the history of mitochondria. All of them are derived from a common ancestor to the exclusion of other bacteria. So we can infer from that that there was one type of event that brought some bacteria inside the cell of a eukaryote to lead to the mitochondrion. Does that basically make sense? Are there questions about this? So again, we're looking at the phylogeny of the genes inside the mitochondrial genome. They group in trees inside the alpha proteobacteria. And they all share a common ancestor with each other to the exclusion of everything else. So mitochondria from humans, mitochondria from slime molds, mitochondria from euglenas, mitochondria from brown algae, mitochondria from plants, they all group in one branch together. So this suggests one evolutionary event in the history of mitochondria. And based upon this pattern that I showed you before, we can in essence hypothesize a single evolutionary event in the common ancestor of eukaryotes. The inheritance of that mitochondria from all of these lineages, and then loss in this one or two branches that don't have them. There's a little cartoon that I've shown you of this sort of evolutionary model. You have bacteria, and then the archaea eukaryote branch. Here's the eukaryote branch, and then somehow a bacteria got inside the eukaryote prior to the common ancestor of all modern eukaryotes. So to help you think about how people think about this, we can draw up sort of what you could call a cell model for evolution rather than building an evolutionary tree. We're going to look at what people think happened in the history of mitochondria. So in general, what they think happened was that there were three main lineages of organisms. The bacteria branched off first, and then there was a common ancestor of archaea and eukaryotes. They split. That's our three-domain rooted tree of life. That's shown down here. And then the eukaryotes started to evolve separately. They got a nucleus. So they packaged their DNA in this membrane-bound compartment. Let's say this is their DNA here. I'm just going to skip ahead. Sorry, so that's their DNA here. They put a compartment around it. So a really important thing that I mentioned related to amoebas, eukaryotes are really good at bringing things inside their cell, wrapping them up in a process called phagocytosis. So people think that this phagocytosis is what happened in the history of eukaryotes. So after the nucleus was formed, this primitive eukaryotic-like cell encountered some alpha proteobacteria. So bacteria had already diversified at this point. There were many lineages of bacteria, including alpha proteobacteria. They became buddies some way, and it brought it inside the cell. It could have been a parasite, could have been a beneficial symbiote, but it got brought inside the cell of that eukaryote because eukaryotes don't have a cell wall around them, at least in their common ancestor. There are some lineages that have separately now invented a new cell wall, but the ancestral eukaryote had this flexible membrane that could bring things inside of it. So it brought inside this proteobacteria. The outside of the bacteria is the cell membrane. That's left over from this enveloping process. So it envelops with its own membrane, and that stays surrounded around the bacteria. We can see this if you look at mitochondria even today. The outside around the mitochondria is a membrane like the outer cell membrane. Inside was a bacterium, probably a gram-negative, because that's what proteobacteria are. So it probably had an outer membrane, a peptidoglycan cell wall, and an inner membrane. It appears to have thrown away some of those components. So maybe thrown away one of the membranes and thrown away the peptidoglycan cell wall. And this is now the mitochondrion. The common ancestor of all eukaryotes basically looked like this. Nuclear DNA packaged inside the nucleus, some membrane-bound compartment with a cell membrane around it containing mitochondrial DNA that is derived from a proteobacteria. Yeah?
When the bacteria first got involved by the bacteria, would you say, or how would we be able to tell if it's a parasite or if it's
Yeah, so it's a great question. How would you be able to tell if this buddy here was a parasite or a beneficial symbiont or what? This probably happened about 1.5 billion years ago. It's very hard to figure out exactly what was going on then. What people have tried to do is figure out what the closest free-living or modern relatives are to the mitochondrion to see what their features are. In some early studies of this, the mitochondria grouped inside a group called the rickettsias. So in the phylogenetic tree I showed, well, somewhere back there, the genes grouped with a group of bacteria inside the alpha proteobacteria called rickettsias. Those are all obligate parasites. So many people for a while hypothesized that this was originally a parasitic interaction. With new data, such as full genome sequences from rickettsias and their relatives, that is no longer supported. So right now, we know that mitochondria came from alpha proteobacteria, but we do not know which branch in the alpha proteobacteria they came from. We don't yet have enough data from other alpha proteobacteria to figure this out yet. There are a lot of people, there was just a paper a week and a half ago about this, very interesting paper, but they concluded that we still need more alpha proteobacterial data to figure this out. So that would be the best way to figure it out, would be to try and see if they sit within a group that is all parasitic, for example. That might answer the question, but we don't yet know. Yeah?
I have a quick question. Is this also a process of endosymbiosis, or is that different?
So this is, as I will talk about in a minute, this is what is generally called endosymbiosis. Endosymbiosis is when a symbiont is brought inside a cell. Symbiont could mean parasite or mutualistic symbiont. It doesn't really matter. But endosymbiosis is whenever some type of partner, a symbiont, is brought inside a cell. There are many examples of endosymbiosis throughout eukaryotic evolution. These two that we're going to talk about today are the biggest. The mitochondrion appears to have been at the common ancestor of all eukaryotes, so that's been important for everyone. As we will see in a minute, the chloroplast appears to be somewhat later, but still very important in the evolution of photosynthesis. But there are endosymbioses in every major lineage of eukaryotes that still continue to happen today. So again, this is sort of the proto-eukaryote. Common ancestor of all eukaryotes looked something like this. And we can then, hypothesize that all of the descendants of that proto-eukaryote looked like this unless they changed something. Like some of them appear to have thrown away the mitochondria. I didn't mention this, but it appears that those lineages that do not have what we would call a mitochondria still have the DNA left over from that mitochondria. It's just moved into their nuclear genome. Yeah?
Do the mitochondria and the chloroplasts replicate themselves, or does this host cell help them?
Yeah, this is an awesome question. So the question is, do they replicate themselves, the mitochondria and chloroplast? So these mitochondria and chloroplast encode some of their own genes, and those genes include some of the components for transcription and translation, but generally not replication. Most of the genes involved in replication are actually encoded in the nuclear genome. This is a very tight partnership in both cases. So you should view a eukaryotic cell as a nucleus and a mitochondrion together.
Yeah.
They're no longer really separate entities.
So then would you say some of the mitochondrial DNA has moved into the nucleus?
Yeah, we're going to talk about this in the next lecture. A lot of the DNA that used to be in the mitochondrial or chloroplast genome has migrated into the nuclear genome of their host. Yeah?
So, chloroplasts and mitochondria don't have circular DNA?
They do. So mitochondrial genome is circular. The chloroplast, most of them, there are a couple of linear mitochondrial genomes. Most chloroplast DNA is circular. But archaeal DNA is circular, bacterial DNA is circular. That doesn't tell us which lineage they came from. But their genomes are in essence very derived versions of these ancestral bacterial genomes that used to be in some free-living organism. Does that make sense? Okay, so this model is sort of, the model for the origin of mitochondria in eukaryotes. An interesting thing is if we look at the genes inside eukaryotic genomes, they actually have, from this current model, two very distinct histories. We have one set of genes that follows that rooted tree of life that I showed you, with archaea and eukaryotes grouping together and bacteria as the outgroup to them. But now we have another set of genes with the mitochondrial genome, where eukaryotes are going to group inside a clade of bacteria, inside the alpha proteobacterial branch. So if we look at, say, the nuclear genome of eukaryotes and the genomes inside archaea and bacteria, so I've drawn in the black bars sort of the species evolution, and these little branches represent different genes inside those organisms. Most of the genes follow this history that we're thinking of in terms of the rooted tree of life that I showed you, with archaea and eukaryotes as sister taxa. And if you built an evolutionary tree of those genes, it would look like this, consistent with that rooted tree, with archaea again and eukaryotes together to the exclusion of bacteria. However, there have been these endosymbiosis events. Some of them appear to have occurred at the base of eukaryotes, like mitochondria. Others, as we will see in a minute, like chloroplasts, appear to have occurred later in eukaryotic evolution. So this creates what you could call chimeras. Eukaryotes have different parts of the cell, have different evolutionary histories. So if you built a tree with some of these other genes, you would see something weird like this. So if the symbiosis was somewhat more recent into some lineages of eukaryotes, you'd have some eukaryotes grouping with bacteria and other eukaryotes grouping with archaea for those genes. It can create very confusing patterns in terms of evolutionary history of genes. And you have to try and decouple this when you're studying the evolution of these organisms because of these distinct trajectories that can happen with the nuclear genome in eukaryotes versus the genomes of some of these symbionts. And that's why, again, with mitochondria, you see that the eukaryotes grouping inside one branch of the bacteria. And we have to use these patterns to try and figure out the complicated evolutionary history of a eukaryotic cell. So again, it looks sort of like this, with different parts of the genome having different histories. And many people use this term chimera, reflecting the ancient Greek myth of this, I forget what it was, a lion, a goat, and something else, all mixed together. That's basically what eukaryotes in fact are, a lineage with many different histories. So that's really important to think about when we now talk about plastid evolution, which is going to get really complicated compared to mitochondrial evolution. So if you look at plastids, again, they look like bacteria. They carry out photosynthesis in many eukaryotes. There are things that look like plastids that don't appear to be photosynthetic in some of the lineages. So the apicomplexans, one of those lineages that we talked about, Plasmodium falciparum, the causative agent of malaria, has a plastid. It's actually related to chloroplasts, but it doesn't carry out photosynthesis. So plastid evolution, if we just do that mapping presence of plastids onto the tree, is much more complex than mitochondrial evolution. We see plastids here in dinoflagellates. We actually see them in a few lineages of apicomplexans. We see them in brown algae, in diatoms, in some haptophytes, but not all of them, in all of these lineages in the Plantae group, in euglenids, in cercozoans, even in some animals. There are these weird sea slugs that have some strange plastid inside of them. But it's scattered, and it's frequently only in one lineage within these big groups. It is not as pervasive as mitochondria. There's not a simple evolutionary model to explain this. Chloroplasts have their own genome, just like mitochondria, so we can start to try and tease apart their evolutionary history, answering questions like, are they all derived from bacteria? Are they all a single evolutionary event? Just like we did with mitochondria. So let's start simple first. If we build an evolutionary tree of chloroplasts, we have to look out for probably 5 or 6 major lineages of bacteria that are candidates for their relatives, because these are the lineages of bacteria that are photosynthetic. If we build evolutionary trees of genes inside chloroplasts, they all group within the cyanobacteria. So that's this lineage here. So one of the major lineages of bacteria, plastids, all group within that branch. They all group together. So if we compare plastids from scattered all across the eukaryotic tree, they all appear to share common ancestry. That suggests a single evolutionary event giving rise to all modern chloroplasts. Again, we had some suggestion that chloroplasts were derived from bacteria from this peptidoglycan data, but that doesn't tell us which lineage they're derived from because all bacteria have peptidoglycan. So again, it looks like they came from somewhere within this one branch, the cyanobacteria. So let's do that same thing with the mitochondria and create a cell model for plastid evolution, in part because plastids are sort of scattered throughout the tree, and in part because there's other data I'll tell you about in a minute. People think that the mitochondrial symbiosis evolved first. And so sometime during evolution, there was another type of interaction. So we start out now with an organism with a nucleus and a mitochondria. It got a buddy. We don't know if this is a parasitic buddy or a mutualistic buddy either. It engaged in some type of interaction with a cyanobacteria. It brought that inside the cell by phagocytosis. And then just like with the mitochondrion, many of these parts were thrown away, although in some lineages, the peptidoglycan cell wall was not thrown away. But some parts were in fact thrown away. So an important term to know is that when an organism brings inside of it a single cell, like bringing inside the mitochondrion, or this mitochondria-containing organism bringing inside a single other cell, the chloroplast, the cyanobacteria, that's called a primary symbiosis. So when you bring inside of you a single thing, the single proteobacterium or the single cyanobacterium, that's called a primary endosymbiotic event, or primary symbiosis. So this is what the primary symbiosis generated with cyanobacteria, the common ancestor, in essence, of all eukaryotes with cyanobacteria. So there's a complication here. So if we go back to the tree, where on the tree are we going to place this single evolutionary event? If we place it at the base of the tree, we have to have many, many, if not hundreds of lineages of eukaryotes losing the chloroplast. What people have figured out through a variety of pieces of information is that the best candidate for where this symbiosis happened is in the branch leading up to the common ancestor of all of these lineages in the plant group. And one reason for that is that all of these organisms here, if you look at the symbionts inside of them, they look like a very simple structure like this. So people have hypothesized, and there's lots of evidence that points to the primary symbiotic event happening on this branch leading up to the common ancestor of plants, or the Plantae group, actually. Okay, so then how do we explain the chloroplasts in these other lineages? This is where it starts to get really bizarre in eukaryotic evolution. So what appears to have happened is, again, the common ancestor of everything had a mitochondrion. This branch brought inside of it a chloroplast. All of the descendants of this branch got plastids. And then these other lineages somehow brought inside of their cell an entire eukaryotic cell that had a chloroplast inside of it. So let's look at what that might have looked like. You have one of these other lineages of eukaryotes that did not yet have a chloroplast. It has a nucleus and a mitochondrion. It then developed a partnership with a photosynthetic eukaryote, enveloped it, and brought it inside, and now you have 5 genomes inside one cell. The original nucleus and mitochondria of this host, and then the nucleus and mitochondria and chloroplast genome of this now secondary symbiont. So if you bring inside of you an organism that already has its own symbionts, that's called a secondary symbiosis. So when people proposed this model, basically people thought they were smoking something. But as people started to sequence DNA from different organisms and characterize the different genomes inside the cells, all of the evidence supports this model. So we can ask, for example, if we take an organism, and I don't know if I have pictures of it, but there are some cells that literally look like this. You can see 2 nuclei inside some organisms, as well as this extra membrane around the second nucleus and the chloroplast. Many of them have thrown away this, what you could view as an extra mitochondrion. Not much use for 2 mitochondria inside a single cell. Many of them have thrown away this second nucleus. Also not necessarily much use for a second nucleus. But there are relics of this hidden inside the genes of all of these organisms. So here's the model again for primary symbiosis here. All of these are descendants of the primary symbiosis. What we can do now is really cool. You can look at the evolutionary relationships among these organisms here, and suppose that this lineage here brought inside of it a red algae. Then the genes in its chloroplast should group in trees specifically with red algal genes, even though its nucleus, its genes in its nucleus, should group with this other lineage of eukaryotes, the kinetoplastids. So what you have to do is look at the difference between the evolutionary trees of the genes in the nucleus and the genes inside the various compartments inside the cell. So in essence, what you're doing is looking for signal if any of these other organisms with a chloroplast, if their chloroplast groups with chlorophytes, that suggests that that cell brought inside of it a chlorophyte, something that was in this branch, and it was brought inside another cell. If their chloroplast group with glaucophytes, it suggests that they brought inside of them a glaucophyte. I'm just representing that. You can sort of cartoon this by coloring the different branches and looking, when I build a tree, if something groups with these different colors.
This sequence shows the
So if we look at euglenas, for example, look inside the nuclear genome of euglenas, it shows that the nucleus is related to kinetoplastids. If we look inside the chloroplast,
this real-time sequence shows a large euglena whose cell wall consists of interlocked.
So in that model, we would hypothesize that this lineage of euglenas brought inside of it a chlorophyte that already had a chloroplast. It was in this Plantae lineage and underwent this secondary symbiotic event. In many of these lineages, like I was saying, you can still see the second nucleus genome. You can build an evolutionary tree of that, and it also groups with chlorophytes. So let's look at diatoms. If we build evolutionary trees of the genes inside the chloroplast in diatoms, they group with red algae. So the model would then be that the diatom branch, some of them brought inside their cell a red algal eukaryotic symbiont. We can do the same thing for all these other lineages. It appears that, red algae were the common ancestor of this branch. These ones may have brought in red algae too. This branch may have brought in green algae. There's all these things scattered throughout the tree of eukaryotes. And again, we're decoupling the evolution, looking at the evolution of each of the individual compartments here to build a model to explain this complicated first primary symbiosis and then secondary symbiosis. Now, I'm sorry to tell you it gets even weirder in some eukaryotic lineages. So it appears that other eukaryotic lineages with a nucleus and a mitochondria brought inside of them a secondary symbiote-containing cell. So this cell had already undergone the primary symbioses and a secondary symbiosis. So now you have an organism that has 7 different lineages inside of it, potentially. Incredibly messy. This is what may be the case for the photosynthetic animals like sea slugs. They appear to have brought inside of them some, I forget which lineage of eukaryote that had a secondary symbiosis. So this is called a tertiary symbiosis. If an organism brings inside of it an organism that already had a secondary symbiosis. And there are some people that have suggested that it has even gotten more complicated. I'm not even going to try and draw some of the other models. But eukaryotes, this is a feature of eukaryotic evolution. The flexible cell structure with a cytoskeleton to maintain the different organelles inside the cell. They bring stuff inside them all the time to eat. Parasites take advantage of this and live inside of our cells. But all sorts of different eukaryotic lineages are bringing inside of them various microbes all the time. The critical thing goes back to, I think it was your question, how does the replication happen for these systems? In some cases, it might just be eating something, and eventually it digests it, and it's gone. But in other cases, it appears that these get eventually stabilized such that it is a permanent symbiosis, and that the replication of all of these different compartments is somehow coordinated, leading to an organism where, many of these different compartments are thrown away over evolutionary time, but in theory, you can have an organism with 7 different evolutionary histories inside of it with this tertiary symbiosis. So we can trace those in the same way that we traced the other ones. I'm not going to go into the exact detail of what those trees look like. It hurts my brain to think about them, so I'm not going to sort of expect people to be able to deconvolute them. But you should be able to think about what trees should look like for primary symbioses and secondary symbioses, and what the different compartments, their trees, can tell you about the history. So we'll stop there.